All right. Okay, so hi, my name is Mauricio. I also work with the team. This talk is going to be the first one that's going to actually go into the tools and talk about the details of what we do with the GATK and our pipeline. As Ami just presented, this is the mapping and duplicate marking step of the pipeline. It is the first step of the pipeline, and it's also the only step, or one of the steps that do not use the GATK at all. So I will be talking about other people's tools here, but most of them come from the BroadE and the Cigar team. So just getting you, placing you in this pipeline that we have, this map of the pipeline. We are at the first step there, mapping and duplicate marking. These will be done right out of the sequencing machine. So the sequencing machine just produces all those short reads, and we need to place them in the genome. We don't know where they go. So the process of placing them in the genome is what we call mapping. And once it's mapped, we also have to find the best alignment for that sequence onto the reference genome. So this is the first of the two steps that I'm going to describe. Here in this presentation, you will see BWA a lot because that's the tool that we use. It's a tool that's developed here at the BroadE by Hung Lee. But nothing is specific to BWA. We do operate with other aligners. It's just that BWA is what we use, and we obviously will be more comfortable with the error modes of BWA than other aligners. But the GATK is aligner agnostic to a certain extent. The second step is to mark the duplicate. So duplication, I will talk in more detail, happens when the sequencer produces multiple reads for the same fragment, for the same actual molecule of DNA. For purposes of SNP calling and all analysis done in the genome, we don't want multiple copies of the same molecule adding to the representation of that site or that haplotype that you're counting, because that will bias all the statistics that we have to do to decide whether or not this is true variation or false variation. So I will get into more detail with that. Those are the two steps that we do. So let's talk about mapping first. I like to call it mapping and alignment. I like to separate the two, although they happen at the same time. Mapping is basically the process of taking these short reads in any order and just trying to find a placement for them in the genome. BWA does that optimally using suffix trees. It's super fast. It's just that we have a lot of reads, so running BWA on a full product of an Illumina run takes a long time. So in here, you're seeing both the mapping and the alignment. The mapping will just find the best position of that short read onto the genome. And then once you find that position, alignment you will try to align it as best as possible. So what happens here, and I'm going to come back to this slide, is that the reads are not perfect matches to the reference. We all have SNPs. We're all different from the reference in many ways. We have regions that just don't match the reference at all, other regions that have a few variants that are kind of making the placement difficult. So in order to complete the alignment, the aligner, BWA, will have two steps. The first one is find the most likely place where this read can be placed, or a few more most likely places. Then try to place it there and align all the bases as best as you can, given all the parameters that you can give it, such as gap open penalty, gap continuation penalty, mismatch penalty, and things like that. So given all these parameters, find the best possible alignment for this read to the reference genome and score it. So there will be a one-number score for that alignment, that particular mapping and alignment. BWA will also try, if the other placements are good, close enough to the one that you tried, it will also try the other ones and see if with a different alignment there, the score is higher. But in general, there will be only one place, and the alignment will just just separate the mismatches and the indels as best as it can. So once it does this, the placement of the, I have a pointer here. I can't see this, I'm colorblind. So the placement of the reads will basically place the reads as you see in that diagram just under the region where it's most likely to be. And then this placement will have a value associated with it that we call the mapping quality. The mapping quality is how likely is this read to be placed here confidently by the aligner. So factors that will lower this confidence are, for example, there are multiple places with pretty good likelihood of matching as well. So regions like very repeated regions will be very difficult to place the short read. Other things are if you're doing a mate paired sequencing and your mate falls somewhere else that is way too far from the location that this read falls, then that's another indication that something may have gone wrong either with this one or the mate. So the mapping quality will try to represent the confidence that the aligner has on placing those reads. So the mapping quality is a very, very important measure for you, or for the statistical machinery of the GATK to identify errors or problems that may have happened in sequencing or just in aligning, or maybe indications that the reference genome may have fuzzy locations in the genome. So going back to the previous slide, I want to point out the Cigar string, which is the diagram here to the right. This is the way that the aligner has to represent the alignment part of this two-step process, the mapping and alignment. Just so you visualize this, the BAM file will not have a sequence with indels marked in it. The sequence that's going to be shown in the BAM file is going to be exactly what the machine read. The machine doesn't read deletions. There's no such thing. It's a concept, not really. There's no deletions in the genome. The machine will read a sequence of letters for us, of nucleotides, and the Cigar string will tell you how to actually place those bases onto the position that the mapping has told you this read goes. So the Cigar string right here says 3M, 1D, 2M, 1I, 1M. This is the letters represent events. So 3M is saying that the three first letters of this sequence are matches. So they will match perfectly the location where the aligner is placing them. D goes for deletions, meaning that there is a deletion between the CAT and the C, right? So in the BAM file, you are only seeing CAT, CT, AG. But by looking at the Cigar string, now you know that there is a deletion, and you should push the rest of the sequence forward after the CAT. Then there are two matches for the CT, and then the Cigar string tells you that the following base is an insertion. So that A, although for the machine it just read as a sequence, it's S relative to the reference genome, it represents an insertion, right? So the Cigar string makes it really simple for you to look at the base, the sequences, and place them to the reference genome, on the reference genome. But the concepts of insertions and deletions are a little complicated to grasp because you don't really see them in the sequence. You have to remember that the sequencing machine really only reads what's in the DNA. Yes?
What's this cigar means?
What the cigar means? I have no idea. Does anybody know what cigar means?
Something about compact gapped alignment format.
Something about compact gapped alignment format.
You guys should ask Bob Hansecker this afternoon. He was one of the creators of spec.
All right. So ask Bob this afternoon. I really don't know what it means, but I know what it does. Okay, so this is how we get mapping and alignment. And one thing that I want to point out, neither of these algorithms take into consideration the base quality score. All of alignment and mapping are done regardless of quality score. The BWA does not look at quality scores. So this is a typical workflow using BWA. Just to familiarize you, since this is not a GATK tool, we're happy to just give you sort of command line so you know how to run these tools. Obviously, I will refer you to their BWA's webpage if you want to actually use this tool. There are many, many parameters. They're useful, especially if you're dealing with different technologies. It's kind of tuned up for Illumina, but if you use it for different technologies, you will have to tweak the parameters. It works very well, but here's how you do it. First, you align it to the reference with BWA align. That's the short read aligner. There's also the long read aligner if your reads are longer than the spec says 200, but I've got good results with 150 base pairs or longer reads. You go to the long aligner, which is just BWA SW instead of align. And the command line is very simple. It's just the reference and your FASTQ file. And the output will be this SAI file. It's just an alignment file. And then you use this SAI file with the BWA SAM-PE. PE meaning paired end. There's also the single end. If you're doing paired end sequencing, you will do that first step twice for both mates. And then you will have two SAI files, as we see in that diagram there. And you can run them both together with SAM-PE to generate the SAM file. SAM file is just the BAM file, but not binary. It's the original alignment format. After you produce the SAM file, you can combine, you have them both combined, and you will go ahead to the post-processing steps that I will talk about later. But this is the general workflow. The one step that's missing here that you only have to do once is to index your reference genome. If you work here at the BroadE, you will already have it indexed in the file system. But if you don't, you'll have to index it yourself. There's a BWA index. You just give it your reference genome, and it will create all the dictionary files that it needs to access the genome efficiently, the reference genome efficiently. So I will go now to marking duplicates, the second step of the pipeline. So like I said, when you sequence, you will produce duplicates. It's just intrinsic of the process. The same molecule will yield many reads. But for us, we really don't want to see that biased representation of molecules. Ideally, we want one good, well-read version of each molecule. That would be the ideal input for the GATK machinery. So what is, in the worst case scenario, things like this can happen. For example, in this diagram, you have the two molecules at the top, and then 1, 2, 3, 4, 5, 6 copies of the same molecule that has just a false positive there, a wrong variant that was just a mistake from the machine. But if you don't mark those duplicates and you try to call a variant on that site, probably you will call that variant because you see it 6 out of 8 reads that you have has that variant. It's just not really true because you're seeing the same molecule that was read over and over again. Maybe that molecule was damaged. There was something wrong with that molecule, and you're just repeating that error over and over again. So when we mark the duplicates, we're not deleting them from the file. We're just saying these are duplicates. For some analysis, do not consider them. For other analysis, it may make sense to look at duplicates. But we mark them all as duplicates, leaving only one true observation of the molecule. And the pileup will then look something like this in the second diagram. And here you see that there will be only one observation of the false positive, and most likely that is not going to be a call with statistical machinery. So that's the goal of marking duplicates. It seems like a very simple process, and it kind of is. It's just looking for things that are exactly the same. And we have many, many tags and markers in the alignment process that will tell you whether or not, actually from the sequencing process that the aligner just passes through, that tells you whether or not these are the same molecule. The easiest one of them being the position where it maps to the genome and the Cigar string. So two reads of the same molecule must be exactly the same. So things that start in the same position and have the same Cigar string are most likely the same molecule. There are other tags that will tell you also if they came from the same lane, if they were read in the same cycle, I'm sorry, not cycle, if they were read in the same run of the machine, that will combined tell you for sure that these are the same molecule, multiple reads of the same molecule. So we don't really do that because Picard has an app for that. It's called Mark Duplicates. It runs It's basically an n-square search. It's going to search for all the molecules and then search it again in the whole file to find duplicates for them and marks them all. We use Picard's mark duplicates to do this step. After, yes?
I have two questions. What's a reasonable duplication rate? And also, why would you want to keep your duplicate molecules?
Okay, for the first question, it really depends on your organism. Okay, so the question was, what is a reasonable duplication rate? And I will forget the second question before I answer this one. It's really dependent on your organism. It's dependent on the machine and your experimental conditions and your sample preparation. There's a lot of parameters that go with what is a reasonable duplication rate. For SNP calling, for most analysis that depend on the statistical significance of the observed basis in the pileup, you really don't want any duplicates at all. You want to have one true observation from each molecule. So you achieve that by marking duplicates. You don't worry so much about the duplication rate coming from the sequencing platform because you will mark them all down. The only problem here is that you're wasting sequencing when you have a lot of duplicates. So for sequencing platform performance measures, it's the higher the duplication rate, the worse it is, right? Now your second question, if you can repeat, I forgot.
Why would you keep duplicates?
Why would I keep duplicates? Okay, good question. So other tools that are not really dependent on that. So let's, for example, let's say you were just trying to see how often the machine makes mistakes in one site, regardless of whether or not the read came from one molecule or the other. You're looking for machine errors, not DNA errors, right? That's a very specific analysis. In that case, it doesn't matter to you if you're seeing the same molecule being read over and over because you just want to see how the machine read that particular molecule or any molecule. You want to see if it always makes a mistake after reading A, C, G, and then after the G, it's always going to read an A, for example. So for that kind of analysis, it really doesn't matter if you're reading copies of the same molecule. But this is a very specific case. Most of the time, you do not want to see molecules, repeated duplicate molecules. So it's not radical to delete to actually remove the duplicates from the BAM file. We just don't do it because it's data and we want to keep it in there. But it's reasonable to do so. Yeah, yeah.
So when you say the same molecule, you mean the molecule bound to the slide when the read happens?
Well, if we're talking about Illumina, you know, the cluster that gets formed when you bind the molecule to the slide gets, there's a small PCR reaction going there, and you multiply it several times, and then you sequence them, right? So multiple copies of the molecule will end up getting sequenced. It happens. There's a huge deal of prep to avoid this from happening, but we still get a lot of duplication. Did you? 1 minute? Okay.
So which copy do you use?
I can't, sorry?
Which copy of the duplicates do you use?
Which copy do we use?
Do you use during variant calling?
It really doesn't matter if they're true duplicates, they will be exactly the same.
Yeah, but the quality, base call quality may be different.
But the base calls will be different?
Base call quality is off.
Oh, I see. Okay. I believe those are defined by parameters in the mark duplicates software. You can probably choose which one you're going to keep. I would assume we keep the highest quality, but I can't confirm that. This would be details on the actual tool. Yeah, I would assume it's the highest quality one, but I'm not so sure. We would have to check the tool. Okay, so quick.
We try to use the GATK
Yeah.
to snippet discovery for RNA-seq data.
Yep.
We also mark duplicates.
Certainly, I think. Let me think about that one. I'll come back to you. I haven't done any RNA-seq, so it's something that I need to think about, but I'll get back to you. Just quickly blowing through this. After marking duplicates, there are two other steps that we have to do typically, and they're just called as part of the mapping and aligning process, which is sorting the BAM file. BAM files have to be sorted, but they come out of the alignment process unsorted. So sorting is very easy. There's an app on Picard that sorts it. It's called SortSem. Or even a BWA can sort it itself if you give it the right parameters. So we get the BAM file sorted. And then after this, we have the most important step on this on this tool, which is adding the read groups. Like Eric said, read groups are key for the GATK to process the information correctly. If it doesn't have a read group, it will barf on you. It will not process your file at all. And no aligner adds the read group by default. You have to add the read groups after the alignment process. So there is a tool for this. Picard has a tool called Add or Replace Read Groups, and you can use it, or you can even use BWA with some quirky parameters to add all the tags of the read group to your file. But this is really important. It needs to be done because for all of the subsequent analysis steps, we need to know which sample these reads came from, lane, platform unit, and all the details that the read group provide to the GATK. Yeah.
Sorting first or mark duplicates first. If we sort it, then it's much easier to find duplicates, right?
Yeah, you sort first, yeah. The typical step is align, sort, add the read groups, and then you mark the duplicates. That's a typical workflow. But I'm tempted to say that it doesn't matter because marked duplicates will probably sort it anyway to look for duplicates. It's just faster if it's already sorted.
I have a question on the read group.
Yep.
So usually, typically, we get a band for each sample. So when you talk about the read groups, do you mean that you actually have to introduce?
Yeah, the sample is one of, so the question is, typically we get one BEM per sample, so why do we need to do the read group thing? The read group, the sample is a big part of the importance of the read group. There are other information in there that is also very important, like which run of the machine was this? Which platform did it come? Is it PacBio? Is it Lumina? Is it Ion Torrent? All of this information is also very important, and sometimes in one BEM file, even though it came from one. It depends on the sequencing center you're talking about. Here at the BroadE, it's all processed for us, so it comes all identified, and per BAM files have their own read groups, and then you combine them all. But you may have one sample that was sequenced over 20 different machines, and you want to combine them all into one BAM file. So you need to add each read group to each one of these runs and then combine them all, right? To combine them all, there's also a Picard tool to combine BAM files. It's pretty straightforward. And it'll combine the read group information as well. So that's, I hope that answers your question. Yeah, so we're out of time. This is just to conclude, I just want to recap what we did here. We aligned and we deduped, and this is where we are in the pipeline. I think the next one will be local indel realignment. Thank you.